we are doing a one-shot revision series. We have completed dark reaction and light reaction in photosynthesis. So we will be completing one-shot revision of light reaction in photosynthesis, and this video is going to be very beneficial for all those who are studying for NEET exam and class 11th and 12th biology exam. Let us do the question answers for photosynthesis. All the questions, this will be a quick revision for you, those who are attending the NEET biology exam and also class 11 and 12 board exams. So this will be a quick revision for you. Let us, in this revision we include dark reaction, light reaction of photosynthesis till Kelvin cycle. So first question is dark, which is the slowest step in photosynthesis. There are two reactions, light reaction and dark reaction. So dark reaction is the slow step in photosynthesis. Now photosynthesis, it takes place where? Photosynthesis takes place in the chloroplast of the cell. In the chloroplast, and photosynthesis takes place in the chloroplast, and chloroplast is present in the mesophyll cell. Photosynthesis takes place in the mesophyll cell, and chloroplast is present inside the mesophyll cell. So photosynthesis takes place in the mesophyll cell and chloroplast is present inside the mesophyll cell. Now next question is light reaction. Light reaction takes place in the thylakoid membrane of chloroplast. Light reaction and dark reaction both take place inside the chloroplast. Light reaction takes place in the thylakoid membrane, and dark reaction takes place in the stroma. Dark reaction takes place in the stroma. Now light reaction is called as the photochemical phase. Light reaction is also known as photochemical phase. Let us pass on to the sixth question. Full form of NADPH, nicotine amide adenine dinucleotide phosphate. Full form of NADPH, nicotine amide adenine dinucleotide phosphate. Now chlorophyll A, the pigment color, the color of pigment chlorophyll A is blue-green. The color of pigment chlorophyll A is blue-green. The color of pigment chlorophyll B is green-yellow. Carotenoid color is yellow, and xanthophyll's carotenoid color is yellow-orange. Carotenoid is present in carrot. Color of carotenoid is yellow-orange and xanthophyll is yellow. So once again, full form of NADPH, nicotine amide adenine dinucleotide phosphate. Chlorophyll A blue-green, chlorophyll B green-yellow, carotenoid's color yellow-orange, xanthophyll yellow color. Now chlorophyll A, it is a lipid molecule. It is a lipid, all the pigments are lipid molecules. So it is water insoluble. Chlorophyll A is water insoluble. Let us see the chemical formula. C55H72O5N4Mg. What is the chemical formula? C55H72O5N4Mg is the chemical formula of chlorophyll A. Chlorophyll A, the structure, it has two parts, porphyrin head and phytol tail. There are two parts for chlorophyll A, porphyrin head and phytol tail. Hello friends, let's carry on the revision. So we have seen in the last question that chlorophyll A, what is the structure of chlorophyll A. Now this is a model, this is a structure of chlorophyll A in which magnesium is present at the center and nitrogen are present at the corners, and this is called a pyrrole. This structure is called a pyrrole. This is the structure of chlorophyll A in which magnesium is present at the center and nitrogen is present at the corners, and this particular structure it is called as pyrrole. So how many pyrroles are there? 1, 2, 3, 4. So there are tetropyrrole in chlorophyll A, tetrapyrrole, and this similar structure is seen in RBC. In RBC, in place of magnesium, iron is present. A similar structure is seen in RBC, red blood cells, and in red blood cells, in place of magnesium, iron is present. In chlorophyll A structure, it is made up of two components, porphyrin head and phytol tail. Porphyrin head is made up of 35 carbon atoms and phytol tail is made up of 20 carbon atoms. So in chlorophyll A, there are 55 carbon atoms. Chlorophyll A is made up of porphyrin head, phytol tail. In porphyrin head there is 35 carbon atom, 5 tall tail 20 carbon atom, altogether 55 carbon atoms. In chlorophyll B, the chemical formula is C55H70O6N4Mg. C55H70O6N4Mg, chlorophyll B's chemical formula. CHO group is present in chlorophyll B. CHO group, it is present in chlorophyll B. Lipid molecules, all the pigments, chlorophyll A, B, xanthophyll, carotenoids, they are lipids. So lipids are water insoluble. In lipids, the amount of carbon atom and oxygen atom is very different. The number of oxygen is very less when compared to carbon atoms. In lipid molecule, the number of oxygen atom is very less when compared to carbon atoms. Whereas in other molecules, the number of carbon and oxygen atoms are same. For example, in glucose, C6H12O6, carbon and oxygen atoms are the same. Whereas in lipid, the number of oxygen atom is very less when compared to carbon atom. Lutein is the yellow color pigment which is present in maize, and it is giving yellow color to maize. And lutein is also present in the eye, and it is present at the yellow spot or the region called macular luteae. Lutein is present in eye, which is the region known as yellow spot or macular luteae, and lutein is also present in maize, and it gives yellow color to maize. It's a pigment. A pigment is a chemical compound which absorbs light energy and excites an electron. Then it gives its electron and energy to another pigment. Another pigment which has less wavelength. So pigment, it is a chemical compound which absorbs light energy and excites an electron, and then it gives its electron and energy to another pigment. So when light energy falls on a pigment, it gets excited, it excites its electron. So light energy is inversely proportional to wavelength. Light energy is inversely proportional to wavelength. So whenever energy increases, wavelength decreases. Whenever energy increases, wavelength decreases, and it is the opposite. Whenever the energy decreases, wavelength increases. Whenever the energy decreases, wavelength increases. Now chlorophyll A, it is called the reaction center, or it is the main pigment. Chlorophyll A is the main pigment, or it is called the reaction center. And antennae is otherwise called the light harvesting complex. Antennae is otherwise called as the light harvesting complex. Antennae is made up of chlorophyll B, carotenoid and xanthophyll. Antennae is made up of chlorophyll B, carotenoid and xanthophyll. All the pigments of the antennae, it fuels its energy to, it gives its energy to chlorophyll A. So chlorophyll A, it has minimum energy and it works at maximum wavelength. Chlorophyll A, it works at minimum energy, it has minimum energy and it works at maximum wavelength. So a pigment will always give energy from to a high wavelength pigment. A pigment will always give its light energy to a high wavelength pigment, and the energy always decreases when it moves from one pigment to another pigment. Chloroplast. A chloroplast consists of double membrane. Chloroplast is the region where all the photosynthesis reactions are taking place. A chloroplast, it consists of double membrane. This is the outer and this is the inner membrane, and this matrix inside the chloroplast, it is known as stroma. Now in the chloroplast, certain granum are present. These are the granum, and this are present as stacks one above the other. So this is altogether called as the granule thylakoid. This is altogether called as the granule thylakoid, and the granule thylakoids, they are joined together by lamellae. This structure is called lamellae or stromal lamellae. So this is the granule thylakoid and this is the stromal lamellae. Photosystem 2 and photosystem 1 are present on the granule thylakoid, the membrane of granule thylakoid, and photosystem 1 alone is present in the stromal lamellae. Now let us study the structure of photosystem. Photosystem, it consists of enzymes. A photosystem, it consists of enzymes. It is made up of the reaction center. The reaction center is the main pigment. It is the main pigment, or it is chlorophyll A. The reaction center is also called as the main pigment. It is also known, it is the chlorophyll A pigment. Now this all together, there are many pigments together, and this, it is known as the Antennae. Antennae is otherwise called as light harvesting complex. Antennae is made up of chlorophyll B, xanthophyll and carotenoids. And this is the primary electron acceptor. So a photosystem is made up of enzymes, chlorophyll A, which is the reaction center, antennae or light harvesting complex, and the primary electron acceptor. Two photosystems, they function inside the thylakoid membrane, photosystem 2 and photosystem 1. If the wavelength of light is more than 680 nanometer, photosystem 2 will not work. If the wavelength of light is more than 680 nanometer, photosystem 2, it will not work. So in photosystem 1, the primary electron acceptor, it is iron sulfide. In photosystem 1, the primary electron acceptor is iron sulfide. In photosystem 2, the primary electron acceptor is pheophytin. In photosystem 1, the primary electron acceptor is FeS, iron sulfide. In photosystem 2, the primary electron acceptor is pheophytin. When a compound gives its electron to another compound, it becomes oxidized. When a compound, it gives its electrons, it loses electrons, it becomes oxidized. On the other hand, when a compound takes electrons or gains electrons, it becomes reduced. Whenever a compound gains electrons, it becomes reduced. So electron transport chain, there is an electron transport chain between photosystem 2 and photosystem 1. When light falls, both the photosystems become active, and there is an electron transport chain between photosystem 2 and photosystem 1 which transports the electron from photosystem 2 to photosystem 1. So this electron transport chain is otherwise called as ETC. It consists of 3 electron carriers. This electron transport chain, it consists of 3 electron carriers, and they are plastoquinone, cytochrome and plastocyanin. So the 3 electron carriers are plastoquinone PQ, cytochrome B6F, plastocyanin PC. Now when light falls, photosystem 2 and photosystem 1 both become active, both excite an electron, and the electron from photosystem 2, it is accepted by primary electron acceptor. The primary electron acceptor of photosystem 2 is pheophytin. From pheophytin, it gives the electron to electron transport chain. Electron transport chain, it has 3 electron carriers. From electron transport chain, the electron moves to photosystem 1. From photosystem 1, the electron is given to the primary electron acceptor, iron sulfide. The iron sulfide, it gives the electron to a compound which is called ferrodexin. The electron is given to a compound called ferrodexin. Ferrodexin gives the electron to NADP+. NADP+ takes that electron and hydrogen, NADP+ plus 2 electron plus 2 hydrogen, and it makes NADPH. NADP+ accepts the electron plus 2 electron plus 2 hydrogen, and it forms NADPH, and this, it is a reduction reaction. Whenever a compound, it accepts electron, it gets reduced, and this is a reduction reaction. Ferrodexin is otherwise called as NADP reductase. Ferrodexin, it is otherwise called as NADP reductase. Photolysis is the breaking of water. Now there is photosystem 2 and photosystem 1. Photosystem 2 has given the electron to photosystem 1, and photosystem 1 has utilized this electron to make NADPH. Now however light falls on photosystem 2, it is electron deficient, so it cannot excite an electron. Now photosystem 2 has certain enzymes, so it will use light energy or light energy to break water. So this breaking of water is called photolysis. Water breaks to form 2 hydrogen ions plus half oxygen plus 2 electron. Water breaks and it gives 2 protons, half O2 plus 2 electron. This electrons, it is accepted by photosystem 2, and this process takes place in the lumen of the thylakoid membrane. This process, it takes place in the lumen of the thylakoid membrane. So oxygen is made in the lumen of the thylakoid membrane, and NADPH, it is made in the stroma. NADPH, it is made inside the stroma.